All right, we are going to spend some time talking about the plasma membrane in a little bit more detail. So in previous lectures, we have kind of gone over the plasma membrane. We've talked about all of these different pieces. So I have this image here now. Remember, the majority of the plasma membrane is made up of our phospholipid bilayer, or phospholipids. You can see the phospholipids in here. This is indicated the phospholipid bilayer. Right here, they're showing the actual phospholipid molecule. Remember, it has the polar head and the non-polar tails. So the polar head is going to interact with the water on either side of the bilayer. So we have the cytoplasm, which is our watery, jelly-type mixture, and then the extracellular fluid on the outside of the cell. And so our polar heads are going to interact with our polar, watery type of areas. And then what we end up having is our hydrophobic region in the middle here. And so this whole region in the inside here is our hydrophobic region. We also talked about the other types of things that we find in the plasma membrane. So we talked about cholesterol, which is labeled here on the left side. So cholesterol, you can see the sterol rings here, the 4 sterol ring chunk of cholesterol. You can see them in several places here. So it is mainly in the area, in the center area there, the lipid, the hydrophobic region, which is where we would find cholesterol since it's a lipid. We also talked about the various types of proteins. So we have an integral protein here. Remember, an integral protein means that it spans both layers of the bilayer, so it's sticking out on the extracellular side, and it's also sticking out into the inside of the cell. Then we have our peripheral proteins. And remember, our peripheral proteins are those that are associated with just one side of the plasma membrane, one side of the bilayer. Then we also talked about glycoproteins and glycolipids. So glycolipid is labeled here. You can see that it has the lipid portion on the bottom, which is, of course, in the hydrophobic layer in the center. And then we have our glycogen type, or sugar chain, that's on the surface of the cell that's going into the watery solution, the extracellular fluid. That's because our carbohydrates are polar, and so they're going to mix with the polar region of the fluid. And then our lipid chains are nonpolar, so they're going to stay in our nonpolar region. Then the other that we have is our glycoprotein. And so our glycoprotein, you can see, is labeled up here kind of toward the top. And so we can see the actual protein that's sticking out of the bilayer, and then we can see the glyco part, which is the carbohydrate piece sticking out there. You can also see on the right side over here, it has labeled the carbohydrate, but this is another glycoprotein, because you can see this is the carbohydrate portion in the extracellular fluid, and then you can see just the top part here of the protein that's embedded in the plasma membrane. So those are all of the different pieces that you should remember of the plasma membrane. And then we remember the structure of the plasma membrane. It's a fluid mosaic model that's semi-permeable. So remember, fluid meaning all of these pieces are kind of moving around on the surface, and that itself is fluid, meaning it can glob out or bud off of the plasma membrane and then can merge back in with the plasma membrane. So it's flexible, it can change shape, things are moving around. And then, of course, it's a mosaic because it has all of these different pieces, mosaic pieces. Now, what I'd like to talk about is I'd like to get into a little bit more detail, specifically about something called the glycocalyx, which includes some of the pieces that we've already talked about. But then what we're going to talk about are how things are moving in and out of this plasma membrane. So we've already talked about how the plasma membrane is this barrier on the outside of the cell. And we mentioned that it's semi-permeable, which means that it allows some things in and out and does not allow other things in or out. And we're going to talk about how that is and how the plasma membrane specifically does that. Before we move into that, though, I do want to mention the glycocalyx, because we talked about the parts of the glycocalyx, but we didn't talk specifically about the glycocalyx. So we have the glycocalyx. So our glycocalyx is the fuzzy coat of carbohydrates that's connected to the proteins and lipids. So what we're talking about are the combination of the glycoproteins and the glycolipids that are on this extracellular, the outside part of the cell. And it makes a fuzzy coat. So the fuzzy coat of carbohydrates connected to proteins and lipids. So namely, this is our glycoproteins. These are our glycoproteins and our glycolipids. So together, the glycoproteins and the glycolipids make what we call the glycocalyx. And the glycocalyx then, as we just mentioned, this kind of fuzzy coat. If we were to look at a cell, let's say that's the surface of the cell, we have all of these glycogen or carbohydrate chains sticking off of the cell. And what this ends up doing is it ends up looking very fuzzy. It makes the cell look very fuzzy on the top when we're looking through a microscope. But it is very fuzzy in that it's kind of like little tiny hairs, all these little carbohydrate hairs sticking off of the surface of the cell here. And these are the glycoproteins or glycolipids, right? So some of them are attached to lipids down here. Some of them are attached to proteins in the plasma membrane. So glycoproteins there, some glycolipids, glycoproteins. So all of these, this right here, this kind of fuzzy area on top of the cell is the glycocalyx. And the function of the glycocalyx, first, the function is for protection. So protection meaning that there's another layer here that something has to get through. So say there's a bacteria that's coming and it wants to get into the cell. It's going to have to try to get through this layer of sticky carbohydrates before it gets to the actual cell and starts damaging the cell. So it's going to protect it. It gives a nice layer of protection. Gives another layer to have to get through. And sometimes the particular bacteria doesn't have a way to actually get through these carbohydrate chains, and so it will stop them from getting into the cell. So protection, and then also adhesion. So adhesion, meaning that this is sticky. So if you think about something that's sugary, if you think about it, if you have a glass of juice and you spill some juice on the counter and then don't notice it, and then later on you come back and you notice something sticky on the counter. That's because, of course, carbohydrates are sticky. This stickiness is going to be seen on these cells. So this stickiness is used for adhesion, so sticking to different things. We can use the glycocalyx to stick one cell to another cell, or for it to be able to stay in a particular location. And then lastly, for cell identification. The glycocalyx actually ends up being a kind of fingerprint for the cell. So the orientation, the way that they're structured, the way that they're in the plasma membrane ends up being a kind of fingerprint, saying that this cell is a cell that belongs in the body, and so then it indicates that this is a cell that shouldn't be destroyed by the immune system. All right, so that was kind of our last identification, or our last piece of the cell that we needed to identify. Now what we're going to do is we're going to be talking about these proteins that are in the plasma membrane. So when we look at the plasma membrane, we have already mentioned that we have these integral proteins and these peripheral proteins. And we actually see, if you look over here on the right side, yellow, we have a peripheral protein here that's on the outer surface of the cell as well. And so we see these different types of proteins. There's the peripheral proteins that are in the inner of the bilayers and the outer of the bilayers, and then we have our integral proteins. Now, in this image, they actually show one of the proteins in a particular function. So in this one, it's a protein channel, and a transport protein. And that's exactly what we're going to be talking about, are the way that we use these proteins in the plasma membrane. So why we have these proteins in the plasma membrane. So first, we will talk about the membrane proteins. So the membrane proteins, many of them act as receptors. So one thing that the proteins do, one, they can act as receptors. And the way that they do this is they'll bind to a signal molecule. So we'll have a molecule being sent out in the body. Let's say it's a hormone, for example. We have a hormone sent out in the body, and then that signal, that hormone signal, is going to attach. So it'll attach to the outside of the cell, and it'll cause a change inside of the cell. So some examples would be different types of hormones, or it could be insulin or glucagon, and then we're going to have that attachment. So a receptor is where we have, where it binds to a signal molecule, to signal molecule outside the cell and cause a change inside the cell. So the way this would look would be if we have our plasma membrane. We have our phospholipids, of course, and then we have our protein. So we have our protein that's embedded in the plasma membrane, and we'll say it looks something like this. So we have our phospholipids, of course, that we continue, and our plasma membrane because the protein is embedded. Okay, so this is our protein here, and this is our receptor protein. This is inside the cell, and this is outside the cell. And then what we have is our little receptor, our little signal. Sorry, this is the receptor. We have our little signal come along. So let's say we have insulin. Insulin is a molecule that's important for getting glucose into cells. So when we eat a meal and we have lots of glucose that we've just eaten, lots of carbohydrates, then our body's going to release insulin in response. So in this case, we would say that this receptor is an insulin receptor. And then we would have something come along. Let's say this is our little insulin molecule, and it's going to fit right in here. And so once this insulin molecule attaches to the receptor, then that's going to cause a chain reaction, or a reaction inside of the cell here. And so when the insulin binds to the receptor, then it's going to actually cause the muscle cell to take in glucose and to use glucose and to store that glucose. So just because we have this insulin attached to this protein, when it attaches to the protein, it tells the cell, through a series of reactions inside of the cell, to allow glucose into the cell, and so that it can use it and then it can store it. So that's an example of a receptor protein in the plasma membrane. Secondly, these membrane proteins can be enzymes. So we've already talked about how proteins, most of proteins are enzymes, and when we have some proteins that are embedded in the plasma membrane, they can be enzymes that are just hanging out and on that surface of the membrane. So these are able to catalyze reactions at the surface of the cell. So we would have a similar type of setup. I'm just going to shorthand these phospholipids. Hopefully that's okay with everybody. And then we have our enzyme, and then my shorthanded phospholipids here. Okay, so then we have our protein here that we are going to say is an enzyme protein. So it's similar to when we already talked about enzymes, right? We have our active site here, just like we talked about before. And this is our enzyme protein that's embedded in the plasma membrane. So for example, two enzymes that we've already talked about, lactase and sucrase, are enzymes that are embedded in the plasma membranes of cells that are in our digestive system. So in our small intestine, toward the end of the small intestine, is where we're going to see breaking down of our disaccharides. Remember, our disaccharides lactose and sucrose are broken down by the enzymes lactase and sucrase. So we can pretend this is either lactase or sucrase. Remember, ACE is indicating that's an enzyme. And then we can say that we are going to have our disaccharide. Of course, this is not the shape of any of those things, but our disaccharide is going to fit right in here. And then this enzyme is going to cleave that disaccharide and make it into our two monosaccharides. And then those monosaccharides can be absorbed in the intestinal cells. So they've been broken right here, and then that allows them to get into the cell. This will actually require some other proteins that would be right here. But for illustrative purposes, we have our enzyme. It's going to assist in breaking those bonds right at the surface of the cell so that both of those things, those monosaccharides, are now available right where they need to be absorbed. The third type of membrane protein that we see are recognition proteins. So recognition type of proteins indicate the type of cell. So indicate the type of cell. So we've already mentioned that we have our glycocalyx that kind of acts as a fingerprint for the cell. But we have specific proteins that are on the outside of our cells. These specific proteins are made by the body very early on. And these proteins are what are going to indicate to the immune system and to every other cell in the body that this is self. So we have self proteins that say that when we have our immune system going along and checking to see if there are any foreign invaders, any bacteria or viruses or anything, it can go along and it's going to attach on to each cell that it interacts with, each cell that it bumps into, and it's going to see if we have these recognition proteins on the cell. So if it attaches to a recognition protein, it says, oh, okay, this is self, you're good, and move on. Then it tries to attach to a cell and look for that self protein, see if it can attach to the recognition protein. And if it doesn't, then it says this is not self and it needs to be destroyed. And it'll go about the process of destroying that. And that's how we would, one of the ways that we get rid of bacteria, for example, is they don't have the same recognition proteins on their cells as we have on our cells. And so these are going to indicate the type of cell, so indicate that it's a self cell. So these allow cells to recognize each other and foreign cells. So as we just mentioned, allows our immune system cells, for example, to recognize every other cell, and then it also allows them to recognize foreign cells because they will have a lack of these receptor or these recognition proteins. So for example, what I've been talking about is the MHC complex. So the MHC complex, or the major histocompatibility complex. So the MHC, this is kind of duplicating complex here, but the MHC or the MHC complex is something that's expressed on cells, and it's unique to each person. So this is the myself indicator. And the myself indicator, the myself protein, is made very, very early on while still in the kind of ball of cell stages inside of the mother's uterus. And so this MHC complex is going to be created to say that this is self and this is self. So it's expressed on cells and is unique to each person. This is what makes transplants difficult because the body is going to go along and it's going to take that tissue that was transplanted into the body and it's going to say this doesn't have the self on it. It doesn't have this MHC on it, the MHC complex, these proteins that are recognition proteins. So that becomes an issue. All right, so the next type of membrane protein that we will talk about are junction or adhesion proteins. All right, so junction or adhesion proteins. So these are going to attach the cell to other cells or structures. Attach cell to other cells or structures. So this would be attaching it to maybe the extracellular matrix that it is creating or to another layer of tissue underneath. So the outside of the cell adhesion molecule attaches to the cell or the structure. And then the inside attaches to the protein network that's inside the cytoskeleton. We've already talked a bit about these because we looked at our cell junctions. Because remember, we had our two cells next to each other, and then we had the proteins in here. Remember the proteins, let's say these are the adhering or the anchoring junctions. The proteins went through the plasma membrane and attached to another one on the other cell, for example. we had these other proteins here, and then this one attached to the cytoskeleton here to anchor the junction. So then these cells were anchored to each other. We saw these in skin cells, for example. So the outside attaches to the other cell or structure. So that would be the outside here of the protein. And then the inside attaches to the cytoskeleton. And that's what we saw here. We have the inside of the protein attached to the cytoskeleton, which is going to be anchoring it inside of the cell, making sure it doesn't move. So for example, dystrophin is a protein, or is a complex. So the dystrophin complex connects muscle cells to surrounding structures. So our muscle cells are going to be moving a lot, right? So when we're moving our leg, moving our arms, all of these things are going to have to be anchored so that they're not just kind of sliding around inside of our body. So we have this dystrophin complex that's going to attach our muscle cells, to the surrounding structures so that even though they're able to move, they're not going to just be sliding around because of not having a connection. So perhaps dystrophin is familiar to you, and this is because if we lose the dystrophin, if we don't have the dystrophin, then we have a disease called muscular dystrophy. And in muscular dystrophy, the muscle cells don't attach very well, and so we have a lack of muscle tone. We have some pain and some issues in the muscles. Then the last type of membrane protein that we're going to talk about are transport membranes. Not transport membranes, transport proteins. So our transport proteins are going to provide an opening to let molecules in and out of the cell. So for example, when we want to get glucose into the cell, glucose is a very large molecule. It doesn't automatically go through the plasma membrane, and we're going to talk about that in more detail. But if we want to get glucose in the cell, because we need glucose in order to have energy, we have a glucose transporter. And so this is actually going to allow glucose into the cell. So if we have our bilayer, of course this is my shorthand for the phospholipid heads and tails, and then we have our protein. The protein is actually going to be a tunnel. So what we're going to see is we're going to allow, so there's a space there. There we go, phospholipids. And then this is going to be a tunnel to allow these molecules to get through and get inside of the cell. So this is our transport protein. And we're going to talk a lot more about different types of transport proteins and why and when we use them and how they work. All right, so before we move into membrane transport, I just want to kind of summarize what we talked about here, because we're going to be now talking about transporting things in and out of the membrane. And this is where we're going to talk more specifically about the semi-permeability of the membrane. So we talked about the different parts of the plasma membrane. We talked about the glycocalyx made up of the glycoproteins and glycolipids. And then we talked about the 5 different types of membrane proteins. So the receptors, the enzymes, the recognition proteins, the junction proteins, and then the transport proteins. And now what we're going to talk about more so are these transport proteins and membrane transport in general, not just those that are utilizing proteins. So remember that our plasma membrane is semi-permeable or selectively permeable. What that means is that some things cross and some things don't. Some things cross it and some don't. So there are different reasons why some things cross it and some things don't. So let's take a look at the reasons why. So first, the size of the molecule. So the smaller molecule can cross a membrane more easily. Right, so if we have a smaller size something, it kind of fit between the cracks of the plasma membrane, so to say. So it can kind of fit between those phospholipids. Remember, all of these things are very fluid. They're kind of moving around. There's some spaces that open up and close and open up and close as we're moving all of these things. So if we have a smaller molecule, then it may be able to fit in between these phospholipids and these proteins and things and can slide right through and get inside of the cell. So a smaller molecule can cross more easily. Whereas if we talk about larger molecules, like for example glucose, which is a rather large molecule, it can't just slide very easily through these through the phospholipids of the plasma membrane. Another aspect is polarity or charge. So if we recall our plasma membrane, right, we have our bilayer. So we have our phospholipids, our heads are on one side, and the phospholipid tails on the other side. Remember that one side, this side, and this side, actually two sides I suppose, but one side of each layer is hydrophilic. Remember, hydrophilic meant water-loving, and that's because these are polar. So this is our polar side at the bottom here, the inside of the cell, and then our polar side on the outside of the cell. And then remember that this central area here is hydrophobic, which means that it is nonpolar. So if we are talking about things that might easily be able to squirm their way through these layers, first of all we have the polar region here, and then we have a polar region here, and then we have a nonpolar region here. So it would have to get through the polar region, the nonpolar region, and then another polar region. So what we actually see is that nonpolar molecules are able to cross more easily. Because even if they're in the water, if we think about oil, for example, if we think about a glass of water and then we dump a little bit of oil on top, the oil may sit up on top of this polar region. But if it is able to interact with, say, some of the cholesterol that's in here, right, our sterol here, it may be able to kind of glob onto the cholesterol, and then it may be able to work its way into this nonpolar region, and then may be able to work its way through this other polar region, maybe through another molecule of cholesterol, for example, or because it's bumping up against some of these other lipids in here. So our nonpolar molecule here is going to have an easier time getting through the plasma membrane than a polar molecule. That's because we have these two regions of nonpolar right next to each other, which makes this region right here very, very thick. So getting something that's very highly charged in this region would be like trying to get water to get into oil or to mix with oil, and that doesn't happen. So our nonpolar molecules are able to cross the membrane more easily, and then the polar or the charged molecules are not able to get past those nonpolar fatty acid tails. This region is just too thick in the middle. Then the third is the presence of a transport protein. So molecules that can't go through the membrane can maybe get through a transport protein then. So they can cross through openings formed by these transport proteins that we just talked about. So we end up having a protein in here, and then it's able to go through kind of like a tunnel. and get into the cell through this transport protein. So it doesn't matter if it is a very, very large molecule, it doesn't matter if it is a polar molecule, it can then hopefully get through because of a transport protein. And there are some things that still can't get in. They have to force their way in, like maybe, say, a bacterium can't go through a transport protein. But many things will have transport proteins if we need them or want them inside of the cell. And they happen to be a large molecule, or they happen to be a charged molecule, and they can't get through the plasma membrane on its own. So let's take a look at different types of membrane transport now. So we talked about what kind of affects things that go through and don't go through. So now what we're going to do is we're going to spend our time talking about membrane transport. So we'll talk about the kind of 3 general types of transport, and then we're going to look closer at each of these types. So the types of membrane transport. So our 3 major types that we'll break down into smaller types are, first we have what's called passive transport. So passive transport is kind of, as its name implies, it's passive. It just happens on its own. What this means is that it does not require energy. So passive transport means that it does not require energy. Things can just move in the direction that they're going to move, and we'll talk about why that happens. But it is able to easily just move to where it's going to go. And then what we're going to talk about is how these things move. They move down their concentration gradient. So they move down their concentration gradient, which means they move from areas of higher concentration to areas of lower concentration. And so we can actually see this when we put, say, like a sugar cube in water. What we'll see is that we're moving from an area of high concentration of sugar, which would be the sugar cube, and moving it to an area of lower concentration, which would be the rest of the water, for example. Of course, this doesn't include a membrane that we're talking about right now, and we'll talk about this in a little bit more detail, but just to give you an idea, what we're doing is we're talking about something going from an area of higher concentration of that molecule to an area of lower concentration. And that does not require energy. Things naturally want to go from an area of higher to lower, to where we're kind of cramming something into one space. If it's allowed to just relax, it's going to spread out. On the other hand, our second type of membrane transport is called active transport. And active transport actually requires energy. And it requires energy because, in this case, it's moving against the concentration gradient, or up the concentration gradient. So move against or up the concentration gradient. Now, in this case, what that means then is it's moving from areas of lower concentration to areas of higher concentration. And that's why this requires energy, because things, unlike passive transport, where things are moving from an area of high concentration to an area of low concentration, like a sugar cube, this would be like taking all of the sugar that's dissolved in a glass of tea or something, and then combining all of that and getting it into a sugar cube shape. And we see that in nature, naturally, that doesn't happen. And so that requires energy. And so we can put energy into the system in order to make this happen, this active transport happen. And we'll take a closer look at some examples of that in a moment. The last type of overall membrane transport is called vesicular transport, or bulk transport. Now, this one, as its name implies, uses a vesicle. So we've talked about vesicles and how they're moved throughout the cell. And in this case, what we're using are these vesicles to transport things. So this transports very large molecules or large amounts of something in a vesicle. So transports large molecules or large amounts in a vesicle. This could also be something that is large, so whether or not it's a molecule or large things. And the reason I say things is, for example, bacteria. Our bodies will bring in bacteria in a vesicle. Bacteria. So when it's trying to get rid of a bacterium, it can grab onto it in a vesicle, bring it into the cell, and then destroy it. So really large molecules, really large things like bacteria, for example, or large amounts of things can be brought in a vesicle. Now let's take a closer look at some of these different types of transport and give some actual examples. So the first one we'll take a look at then are different types of passive transport. The first type of passive transport that we are going to talk about is diffusion. So diffusion is where we have small, relatively nonpolar molecules. Remember, nonpolar molecules can get through that plasma membrane. So this is where we have our small, relatively nonpolar molecules can go through the membrane on their own. And so this is a diffusion or a movement across the membrane on its own. This doesn't require any facilitating. And this is because we're talking about passive transport. So actually, let me kind of open this up again here just to remind us that we're talking about passive transport. And remember, passive transport means no energy. So no energy is required. And the first type of passive transport we're going to talk about is diffusion. So again, this is where we have our small, relatively nonpolar molecules go through the membrane on their own. And when we talk about things going through the membrane on their own, and we're talking about passive transport, remember we're talking about things moving from areas of high concentration to areas of low concentration. So again, we're moving down the concentration gradient. So if we kind of take a look at this, what we might have, let's say we have our plasma membrane, right? So my shorthanded version of the plasma membrane here. These are our phospholipids with our fatty acid tails, which aren't just straight like this, but for ease will be straight. Phospholipid heads here. So we have our plasma membrane, and what we're talking about are things that are able to diffuse, right? And what we talked about, if I draw this over here on the side here, diffusion is not just as far as the plasma membrane is concerned. When we talked about our glass of water that we had with our sugar cube, when we put the sugar cube in, and we had our glass of water, what we saw was diffusion. It doesn't have to do with a plasma membrane per se. We're talking about it through the plasma membrane, but diffusion is what we see when we add a sugar cube into water, and then things move from an area of high concentration to low concentration, and it doesn't require any energy. So we end up seeing the sugar kind of evenly dispersed throughout the water. Same thing with salt. If you put some salt in the water, you have a huge lump of salt, and then you stir it with a spoon, and then that's going to move throughout the solution, dissolve in solution, and it's going to be relatively equal all over the place. Now, when we talk about diffusion, when we talk about the movement of molecules, things want to be equal. So things are going to move down the concentration gradient until concentrations are equal. Whoa. So the reason I capitalize equal is because that's typically how I like to explain these types of things. I think it makes it easier to understand that what we're trying to do is things want things to be equal all over the place. So the sugar cube starts out as a high concentration sugar cube in the center, but it's going to spread out, and it wants to be equal, so it actually spreads out pretty evenly. And we know that because if we take a sip of it, the sip at the beginning is pretty close to the same as the sip at the end, unless, of course, you've oversaturated it and you've got sugar sitting on the bottom. But if you don't do that, if you put a small amount of sugar in something, the first sip of it and the last sip of it are going to taste relatively the same because the sugar has dispersed and it's made it equal. Same thing if you put salt water in there or salt in there. And we see the same thing then with our semi-permeable membrane here. So if we had a molecule that's in the outside of the cell here, and say it's relatively nonpolar, again, maybe it's very, very small, relatively nonpolar, and we have lots and lots and lots of this here, right? So lots and lots and lots of whatever this is. Let's say it's oxygen in the lungs. So these are little oxygen molecules. We've just breathed in a nice big breath of fresh air. So we have lots and lots of oxygen in our lungs. Now we're going to get this oxygen from our lungs into the bloodstream. In order to do that, we're going to have diffusion occur, and which means is that our oxygen is going to move through the plasma membrane to an area of high concentration in the lungs, right? In the actual like air sacs of the lungs, the holes of the lungs where we're breathing in, now has a high amount of oxygen in it. And then we're going to try to get that across this plasma membrane into the cell to get it to the bloodstream, right? Because we want to get into the bloodstream so that we can take that oxygen and bring it all over the body and feed all of the cells of our body with oxygen. So we breathe in the oxygen, which means in the lungs we have a high concentration of oxygen because we just took a big deep breath in, lots and lots of oxygen. If we're breathing in, that means that we have very little amounts of oxygen in the cells that are going to lead to the bloodstream. So you can see we have a high concentration up here and a low concentration down here. And so we're going to see that the oxygen is going to move through the plasma membrane, going from an area of high concentration to an area of low concentration. And so then what's going to happen is all of this is going to continue moving across here, oxygen by oxygen by oxygen. All of these are going to move across here until we have about the same amount of oxygen on this side as we have on the other side, because it wants to make it equal. It wants to have this one side of the semi-permeable membrane and the other side of the semi-permeable membrane have about the same concentration. So right now I'd say maybe it's about the same on both sides. And so then at this point, there's no net movement. What I mean by net movement is a large amount moving across the plasma membrane in this case to get to the other side. Now there's still some movement. We'll have some moving this way, some moving that way, but there's no large change in movement, no net movement, meaning they're not all going in one direction. Once it's equal on both sides, then we'll just have this kind of flowing back and forth of these different molecules because they're able to move back and forth, but no change, none of this going all in one direction and making it more concentrated on one side compared to the other side. And then we would see the opposite, just as a side note, we would see the same thing occurring but with CO2. If we saw CO2, we would have lots of CO2 in the blood over here, and then when we're breathing in, we're trying to get rid of the CO2, so we would see the CO2 then diffuse across the plasma membrane and get into the lungs so that then we can breathe out the CO2. But we have a high amount of CO2 in the bloodstream, and then we would have a low amount of CO2 in the lungs when we breathe in fresh air. And then we have this exchange occur to where we get rid of the CO2, put it into the lungs via diffusion, and then we take the oxygen and get it into the bloodstream or into the cells that will then go to the bloodstream, and that's also diffusion. So the first type of passive transport that we see is diffusion. Now the second type of passive transport that we're going to talk about is called facilitated diffusion. So facilitated diffusion is very similar to diffusion, but has one big change. So in this case, we are going to allow larger or charged molecules. to diffuse across the membrane. So in this case, what we're doing is we are having to facilitate this. And what that means is that these things are larger, and they're charged, or they're polar, so they can't get through all by themselves. So instead, we have proteins that are going to be in here, and they're going to facilitate diffusion. Right, so we have our plasma membrane here, my simple shorthand drawing of the plasma membrane, and then this right here, this is a protein channel, also known as a transport protein. Remember, we talked about that already. So transport protein. So in this case, we're still talking about diffusion, and since we're still talking about diffusion, that means that we are moving molecules down their concentration gradient, meaning moving from areas of high concentration to areas of low concentration. So we're still having diffusion. It still does not require energy, and we still want to get them to equal. Now in this case, now we are requiring a transport protein to form an opening in the plasma membrane. So the transport protein forms an opening for the large molecule or charged molecule to travel through. So in this case, we might say that this would be glucose, for example. So I'm not going to draw all of these. Glucose is a six-sided, so it's a hexagon. I'm just going to draw circles to make it really easy. So these are going to be glucose molecules. So we have just eaten a big meal. Let's say we just ate a lot of potatoes. And so we've gotten through, we're in our digestive system, the latter end of our small intestine, and now we have lots and lots and lots of glucose in our small intestine. Now glucose is too big to fit through the plasma membrane. It also happens to be polar. It's charged, right? So it's polar and it's large. I'm going to say these are glucose. Now we have lots and lots and lots in our intestines, and we don't have that much glucose in our actual cells. We do have some, of course, because we use glucose to make energy, so the cell has some glucose in it, but we have by far a lot more in the intestines because we just ate a high glucose meal. So in order to get the glucose into the cells, which is where we need it in order to use it to make ATP and have energy, then we're going to see the use of this transport protein. Now the transport protein is just like an empty channel or an empty tunnel. So it can be open, and it can just allow things to flow through it. So when there's a high concentration of glucose, that glucose is just going to easily flow through from the area of high concentration to an area of low concentration. And so this isn't going to require any energy at all. It's just allowing for the space to get through, because again, it's too large of a molecule to be able to get through. Now, when I'm talking about glucose specifically, I actually want to kind of change my statement a little bit in the fact that when we're talking about glucose, as I use this as an example specifically, it's not just a tunnel that's going to go through. Lots of things can just use a tunnel, and it can go right through, and it's still facilitated diffusion because it doesn't require energy. Now, we can talk about glucose, and then it has a specific carrier protein. So let me kind of zoom up here. It has a carrier protein. And this carrier protein, what that means is that it's going to bind to the molecule, change its shape, the protein changes shape, and then it releases it on the other side. So it binds to the molecule, the protein changes shape, and then releases the molecule on the other side. So carrier proteins are specific. So they have to have a particular binding site. So it has to bind the very specific molecule. So it's just like the active site in enzymes, just just like the active site of enzymes, where it fits like a key in a lock. So if something else is in high concentration outside of the cell and it's bumping into this carrier protein, if it's not the correct carrier protein for that molecule, then it's not going to allow it to go through. So if we look up here in the illustration again, we would have something like this. And then our glucose molecule, and we have our high concentration of glucose out here, is going to fit into this kind of active site here. And this part up at the top, I could probably do a better job here with the drawing. So this is our active site here, our receptor site. So we have our glucose molecule. It's going to go in here. The side that is toward the intestine, as our example, or the outside of the cell, is open at the moment. Then once the glucose sits in there, then what's going to happen is this top part is going to close off, and then the bottom part is going to open and allow the glucose to come out. So this part closes off, and then this bottom part opens up. And when this bottom part opens up, then that allows the space for the glucose to now come out. Now it's moved to the other side of the cell. And so this will happen over and over and over and over again. And so we have glucose transporters that transport the glucose into the cells just in this way, but it won't transport other things. So for example, we remember that glucose has isomers. It has the isomers galactose and fructose. And they will not transport galactose or fructose. So it's specific to that molecule, even if it is an isomer where it looks very, very close. So these are different types of facilitated diffusion. So in this example, so we're talking about facilitated diffusion. We're using something that's going to facilitate the diffusion, right? So the moving of a molecule from the area of high concentration to low concentration. One way that that happens is through this carrier protein. So where it binds the molecule and then the protein changes shape and it releases it on the other side. But we also have ion channels. So these ion channels are going to open a tunnel for the ions to flow across the membrane. And so these were the ones where I was kind of talking about how they're more like a tunnel, or more just like an open space for things to flow through. They're not always open. Sometimes they're closed. We have gated channels that will open and close depending on something. But sometimes then they're just always open, and they're always moving ions across the membrane from the area of high concentration to an area of low concentration. So again, we're still talking about diffusion, high concentration to low concentration. We're just talking about things that are facilitating diffusion now. So for example, we have a calcium ion channel. So our calcium channels in our muscles allow for the calcium to flow into the muscle cells, which causes a muscle contraction. So we'll have these channels, and just like our other illustration, let's say this is our protein, and then we have this channel that is going to allow the ion to go through this protein in the plasma membrane. I think we can just assume now that we're in the plasma membrane, and this is our protein. So we actually have the ability to have gates on this ion channel. So then we have the gates, and in this case, if we're talking about calcium channels, and we're talking about muscle contraction. Calcium is necessary for the muscles to contract. So when we have high amounts of calcium, it sets off a series of things that is going to allow for contraction of a muscle. Now in this case, we're talking about gated ion channels. So this gated ion channel can open or close in response to a signal. So the signal, it depends on where this gated channel is and what its function is. But some of the signals could be a change in charge. It could be a change in the pH. It could be a change in the temperature, in tension, etc. So we could have various changes that are then going to trigger this gate to open up and allow things to flow through. So for example, with our calcium channels, they are usually closed. So we usually have these calcium channels closed, or these gates closed. But then when we send a message, so we send a nerve signal to that gate, then it's going to be opened. So we can kind of pretend we're sending a nerve signal here. Basically what we're saying is, I want to move my leg, for example. You've decided you're going to stand up from your chair and walk. And so you're sending a message from your brain to your leg muscles to do that. And so then when we do that, that message is sent to that gated channel. Usually it does this by a signaling molecule, so it actually sends a molecule. When that happens, then this gate is going to open up, get out of the way, and then we're able to get our calcium ions in there. So we've got calcium hanging out outside here. Remember, we're still talking about diffusion, which means we have a high amount of calcium out here in the outside of the cell, plasma membrane, and we have low amounts of calcium inside of the cell. So we open up our gate, then the calcium is allowed to flow through this channel and get to the other side, to an area of lower concentration. And again, it wants it to be equal, so it's going to continue flowing across that channel until it gets to equal. In this case, when we're talking about muscle contraction, then at some point we stop sending that message here, and then this gate is going to close again. And then when the gate closes, we're not using that muscle for contraction at the moment, and then we're going to stop those calcium ions from moving across the plasma membrane. So the next type of passive transport I'd like to talk about. So we've talked about diffusion, right? These are for small, relatively nonpolar molecules. Now we've talked about facilitated diffusion, so we have to facilitate it in some way using a protein, and that allows us to move larger or charged molecules. We've done that in two ways, through carrier protein or ion channel. So we're still talking about proteins that are embedded in the plasma membrane. We're just talking about how some of them have a specific active site, like the glucose transporters. Or we can talk about ion channels that are gated. So we have our gated ion channels. Now the last type of passive transport that I'd like to talk about is called osmosis. And when we talk about osmosis, rather than talking about these different molecules that we talked about, like calcium moving across the plasma membrane, or talking about glucose moving across the plasma membrane, what we're talking about is the diffusion of water. So the diffusion of water across a membrane. To areas of high solute concentration. So the key here is that I said high solute concentration. So when we're talking about osmosis, we're going to be taking a look at solutes versus the water itself. So if we remember, a solute is something that's dissolved in a solvent. So the solute would be different molecules, right? Different molecules or ions. The solvent then is water, right? So water is the liquid, and then we have these things floating around, these molecules, these ions, which are the solutes. So when we have our plasma membrane, this is the bilayer here. Now I'm getting even more simplistic here. So our plasma membrane is here. We have lots and lots of water up here, lots and lots of water down here. However, when we talk about different types of solutes, let's say inside of the cell, we just ate a big meal, lots of salts, lots of glucose. So all of these things have been moving into our cells, right? So we've utilized these glucose transporters. We've been moving glucose into the cell. We have our ion channels. We've been moving sodium and chloride into the cell. And right now our cells are really, really filled with all of these different solutes. And our digestive system is relatively empty of these solutes. because we've moved so many of them over into the cells because we've been going through that process. So our water movement is actually going to move toward the high concentration of solutes. So water moves from an area of low solute concentration to an area of high solute concentration. And this is because water is attracted to the solutes. And that's because our solutes will have a positive charge or a negative charge, and we have our polar H2O. Remember, we have that partial positive and the partial negative. And so they're going to be attracted to the solutes, which may be either positive, which is attracting the negative, or negative, which is attracting the positive. And so our water molecules are going to be moving toward the solutes. And the way that I often think about it is that what we're trying to do, in the same way that we want both sides of the plasma membrane to have the same amount of glucose, for example, and so we have diffusion moving from an area of high concentration to lower to make the glucose even on both sides, we want to have these solutes, whatever these are, these green ones, the glucose, the sodium, the chloride, we want to have those be in the same concentration on the inside of the cell as in the outside of the cell. In order to do that, instead of moving the solutes out of the cell in this direction, because we don't want them out of the cell, instead what we're doing is we're moving water into the cell so we can dilute the contents in here, so we can dilute all of these solutes to make it the same concentration inside of the cell as outside of the cell. Right, so if we had a glass that we have put on the counter and we have a glass next to it and we have water in both of them, the same amount of water. If we dump, a half a cup of salt in one of the glasses, it's going to have lots and lots and lots of salt. If we dump, a teaspoon of salt in the other glass, it has a lot less. Now, if we had a membrane going between the two instead of the glasses themselves, what we would see is that the water is going to be moving toward the glass, or toward the side of the membrane now, that has the high amount of salt. And it wants to dilute that high amount of salt to make it even on both sides, to make the same amount of salt water concentration on both sides of the plasma membrane. So the amount of a solute in a solution affects the movement of the water in and out of the cell. So where we have a high concentration of solutes, we're going to see a high movement of water. And this is actually dependent on the concentration as well, on how fast this happens. So if we're looking at two sides of the plasma membrane and we have, let's make up some imaginary number, like 10 items over here, and we have 9 items over here, the movement of water is going to be relatively slow over here to try to make it equal, 9.5 on both sides. However, if we have our plasma membrane and we have 100 items, whatever that means, over here, and we had like 2 items over here, we're going to see a high amount of water movement very, very quickly trying to dissolve the 100 items to make them even on both sides. The concentration. So again, items and these numbers are silly numbers, but to try to get the idea across. Now, one last thing, my drawing is a little bit misleading here because water is a polar molecule, and since it is a polar molecule, it's not just going to very easily move across the plasma membrane. Now, some water does just move across the plasma membrane, but it is polar, so it doesn't cross very easily. And since it doesn't cross very easily, what we actually have are called aquaporins. So aquaporins are actually channel proteins that allow water to move across the plasma membrane. So channel proteins. And these are just for water. Hence the name aqua, meaning water, and then porins. They're pores, or holes for water to go through. And that's what I drew here, are these little holes for water to go through. And so that's going to allow water to move back and forth however it needs to move, depending on the concentration of solutes. And that can change over time, because down here, all of this stuff we just ate and got into the cell, let's say, is getting transported into the blood to move throughout the body. Then that's going to decrease the concentration of these items here, and then maybe up here is going to be in a higher concentration now. And if that's the case, then the water is going to start to move back the other direction to try to dilute the contents in the intestine. And then back and forth, depending on where the solutes are. Now, when we talk about osmosis, there are three key terms that you should be familiar with. And they are hypertonic, isotonic, and hypotonic. So hyper means greater than or more. Iso means same or equal. And hypo is less than. And when we're talking about tonic, we're talking about the tonicity of something, meaning basically the contents of something without getting into the terminology there. So basically the contents. So for example, if we talk about an animal cell, remember, let's bring this back to our cells. If we talk about an animal cell, let's talk about a blood cell. If we have a blood cell, and let's say we put it into a solution. We have our blood cell, and we put it into a solution. If that solution is hypertonic, what that means is that it has more solutes in the solution, because it is hyper, which means more solutes in the solution than there is inside of the cell. So we're placing our cell in a hypertonic solution. Now, if we put that same blood cell in our isotonic solution, what we're then saying is it has the same amount of solutes. Right? So same blood cell in here. Let's see if we can make it about the same amount of solutes inside the cell as outside of the cell. This would be an isotonic solution. And then hypotonic, as you can imagine, would mean that there are less solutes in the solution than there are in the cell. So we have more solutes in the cell and less outside of the cell. So this would be a hypotonic solution that this cell is in. So now when we look at this, remember that our plasma membrane is semi-permeable. And what that means is things can move in and out, right? So one of the things that we just talked about can move in and out very easily through these aquaporins is water. And so when we talk about hypertonic, isotonic, and hypotonic, we're talking about the solution that we put a cell in, and then we're going to be talking about the water movement. So if we are in a hypertonic solution, this first container, and we put a cell in there, and there's water in the cell, and we're in a solution that has the solvent of water, which direction do you think water is going to move? Do you think it's going to move inside of the cell, toward the inside of the cell, or do you think water is going to move from the inside of the cell toward the outside of the cell? So remember that water moves to or toward areas of high solute concentration. So what we would see then is lots of water movement out of the cell. So our water is going to be leaving the cell and trying to dilute the contents of the solution. And in this case, when this happens, when we place a blood cell in a hypertonic solution, we are going to lose so much water out of the cell that it actually crenates. So the cell crenates, and it actually ends up being this kind of spiky looking little tiny blood cell. It has crenated because it's basically shrunk down. It's lost all of its water, and it's just this plasma membrane with very little water on the inside because of the solute concentration and our osmosis, our water moving. Now if we move into the next situation, our isotonic situation, are we going to see water moving in toward the cell, or are we going to see water moving from the cell out toward the solution? We are going to see both. In an isotonic situation, we're going to see water moving out of the cell as well as water moving in the cell. However, it's about equal. So this water is going to move through those pores, back and forth through those pores, but they're going to move back and forth about equally. So it's not going to change the shape of the cell because we're moving in and out. The amount of water, it basically maintains the same amount of water. What this means is there is no net water movement. In this case, when we had crenation, our water movement, our net water movement was out of the cell. Over here, we have no net water movement, so the cell stays the same, and we have about equal moving back and forth. Now, how about our hypotonic solution? Are we going to see water moving in to the inside of the cell, or are we going to see water moving toward the outside of the cell? Right, we're going to see a lot of water moving into the cell because, again, it's trying to dilute the contents of the cell. It's trying to dilute those solutes. And since there's so many solutes in the cell compared to out of the cell, we're going to see lots and lots and lots of movement of water into the cell, so much so that it actually is going to burst the cell. So the cell bursts, and all of its contents are going to come falling out because it cannot withstand the pressure. It cannot withstand the osmotic pressure, which is the pressure from the water moving in toward the solutes. So it actually bursts the cell, and so in this case, we have our net water movement is going to be into the cell. And then we burst the cells. So if we take a look at this, we can talk about this in various ways. We can talk about this in regards to bacteria, and we can talk about this in regards to plant cells as well. However, if we recall, bacteria as well as plant cells have a cell wall. And so we don't see quite the same thing happening with plant cells and bacteria cells. We have the cell wall on the outside, and then we have the plasma membrane. So if we're pretending that this is a plant cell, our very quick drawing of that in a hypertonic solution, we're still going to see the net water movement out, just like we saw in the animal cell, the blood cell. But instead, what we're going to see is the plasma membrane is going to get kind of spiky, like we saw with the red blood cell, but we still maintain the cell wall out here. And since we still maintain, or the plant still maintains the cell wall out there, it maintains its shape, and it doesn't crenate too much. It actually still is attached to the cell wall. This is called plasmolysis. So we have shrunk down this cell, and it's still kind of attached to the cell wall, so it's okay, but it's lost a lot of its water. In an isotonic solution, we're going to see the same thing. The cell is going to be fine because we see the same amount of water movement in as out of the cell. And then on this side, I'm going to draw in a hypotonic solution. So if we have our plant cell here with its cell wall, and in this case, we have our net water movement in the cell. And in this case, rather than us having cell lysis, which I didn't sell here, cell lysis, which is a bursting of the cell, instead we're going to fill this all the way up to where the plasma membrane is going to basically be right up against that cell wall. And so it's going to be as full as it can get filled with solution. And in this case, in a plant cell, it doesn't actually burst. It doesn't go through lysis. Because it has that cell wall for protection. So it's going to maintain it. The water will still try to move in, but just as much is going to be moving out because it's at the cell wall. It's at capacity. So you should know these three types of solution, and this is very important. So for example, if we think about a clinical application for blood cells, for example, this is why we do not give people just plain water or clean water in an IV bag. It has to be saline solution. The reasoning is if we put plain water in the bloodstream in the body, then that would be a hypotonic situation. We would be putting something with a lower solute concentration in the bloodstream, and then that would cause the water to rush into our cells and burst them. If everybody's blood cells are going to be bursting, then that would be very, very dangerous. Instead, we put saline solution, and that's because saline solution is an isotonic solution to our blood and to our cells. And so when we put isotonic solution in there, then we have this equal amount of movement of water back and forth, and then our cells are fine.